Hello everyone. We are now going to go over the bone marrow flow cytometry from a 65-year-old man who presents with skin lesions and leukocytosis. So let's start with CD45 and side scatter. And here we see there are cells in the lymphocyte gate as defined by bright CD45 and low side scatter. And there are some cells in the monocyte gate which has CD45 bright expression and slightly higher side scatter than the lymphocytes. And here we have a large number of cells in the blast gate, which is defined as a gate with dim CD45 expression and low side scatter. However, here we do have a slightly higher side scatter than we normally see in blasts. And there are few granulocytes in the specimen also. And when we follow this gated cells, they express no CD34 or CD13. They are positive for CD38. And they are positive for CD7, pretty bright, and they're negative for CD19. 19 is a lineage-specific marker for B cells, and here again they're positive for CD7, but they're negative for CD2. And there are some add-ons that we can see that in the next tube, CD123 versus side scattered, we have gated just the 123 positive cells, and it looks like these cells have CD38, just like what we saw in our blast population, and they are HLA-DR and 123 positive. They have partial CD4 expression, and they're negative for CD117. They have bright CD56 expression, and they're positive for CD303, and they're negative for CD64. They're negative for CD22, and they're negative for MPO. So now let's see what they are positive for. They're positive for CD123, CD56, CD7, partial CD4, HLA-DR, and they are positive for CD303, and they are negative for MPO. So myeloid specific markers are negative, and they're negative for B cell markers CD19 and CD22. They were positive for CD7, but they are positive for other T cell markers like CD3 and CD2, but that's not shown here. CD2 is shown here, but other T cell markers are not shown here. And they're negative for myeloid markers CD13, CD33, and markers, monocytic markers like CD64, 11B, etc. So not everything is shown here, but they were negative for all other lineage specific markers. So when we have a population of blasts that is negative for lineage-specific markers of myeloid and lymphoid lineage, and they are positive for 123, 303, 56, 4, then we can call it as blastic plasmacytoid dendritic cell neoplasm. Blastic plasmacytoid dendritic cell neoplasms, or Bpdcn, are very aggressive neoplasms, and they do present with skin lesions, and they have a lot of blasts in the bone marrow and the peripheral blood, and they can look somewhat plasmacytoid. And these arise from the PDCs. PDCs are the plasmacytoid dendritic cells, and the specific markers are 123, 303, and some labs perform TCL1, which is also TCL1A, which is also a specific marker for PDCs. And they're often positive for CD4 and CD56. Other lineage markers can be positive. Most commonly CD7, CD33 can be positive. Sometimes markers associated with T cell lineage like CD2, CD5, sometimes even CD13, 79A, all these can be positive. But they'll be negative for cytoplasmic CD3, CD19, MPO, all those lineage specific markers. One thing you have to remember is CD123 has to be positive. And CD123 is not positive only in PDCs. Actually, even myeloid leukemias will often have CD123 expression, especially the monocytic lineage ones will often have CD123 expression. So 123, 4, and 56. This is the combination that most labs use. Not every lab uses CD303. Very rarely they use that. These are the most common markers they use are 123, 4, and 56. And if it is positive for all three, then and negative for other lineage specific markers, we can call it Bpdcn. But individually, none of these are specific for PDCs because 123 can be positive in myeloid cells. 56 again can be positive in a lot of myeloid neoplasms. 4 again can be positive in AMLs and monocytic neoplasms. So that's why they do rule out monocytic lineage. Like for example, here we see that these are clearly CD64 negative. So you need to make sure you don't overcall something as Bpdcn just because it expresses CD123. And normal PDCs that we can see a small population of normal PDCs in normal bone marrow flow cytometry, and those are 123 positive and HLA-DR positive. So here the neoplastic cells are also HLA-DR positive. And if you remember, in the myeloid maturation, we would have seen how HLA-DR distinguishes between PDCs and basophils. So here, that is a good thing to remember because PDCs are positive for HLA-DR, which we see here. So the take-home point is Bpdcns are very rare, they're very aggressive, and they're positive for CD123, 56, and 4. And when you see this combination in the absence of other lineage-specific markers for myeloid or lymphoid or erythroid or megakaryocytic, then you can call it as Bpdcn. And some labs use other plasmacytogenetic cell markers like 303 and TCL1A. Those are very more specific. And if you see that, then you can call it Bpdcn also. Thank you.